All right. I don't know. I didn't think they were too bad. All right, so let's do it. So question one. Question one was about the effect of caffeine on bees, right? So it says in flowering plants, pollinators, pollination is a process that leads to fertilization, yada yada yada. And question A says on the axis provided, construct an appropriately labeled graph to illustrate the effect of caffeine on probability of bees revisiting a nectar source. Okay, so we've got this graph here, and I've just gone through and sort of copied over the data that they gave us. So the data that was on the first page here, and I've included the error bars because I felt like the error bars were pretty important on this. Because the error bars allow you to determine whether or not the data is significant. Okay, so the first question, question A, is just about constructing the graph, right? So I think that this is probably worth 2 points, constructing the graph. 1 point for having the correct labels and scale and title and everything like that, and 1 point for having the stuff on the graph, so having bars with the error bars. I'm pretty sure that you need error bars for this, but I guess it's possible that you don't. But I'm pretty positive they were going to require you to do error bars.
Did you need to label your axes?
Oh yeah, I didn't label the other axis, did I? Thank you for that. So this was probability of bees returning.
Wait, were you actually supposed to put the full bar in there? Or if you just put the point and then the error bars?
Probably the full bar. Yeah, a bar graph makes the most sense in this case because these are treatments. These are flower type and time period. Something like that. You don't necessarily have to have this label here because these are distinct times that they've given you, and the labels for that are in here. But you definitely need to have the probability of bees returning on this axis. Thanks for that. Okay, so question B said, based on the results, describe the effect of caffeine on each of the following. The short-term period. So we can see that because the short-term period has the error bars overlap, that means that there's no effect, that there's no significant effect on the short-term bees activity. The long-term memory of the nectar source, however, we can see that there's no overlap there. And so that would indicate that caffeine increases the bee's ability to remember a nectar source. Yeah.
Just for bar graphs in general, like you can, because I had to follow like the x-axis, so I had like 10 minutes like way over here, and then I had 24 hours way over there.
Yeah, you didn't need to do that. I don't know if they'll. Did you still do a bar graph?
So then I like crossed out my y-axis, like the probability of these returning. So I have to really make this
Oh yeah.
do a new graph in the middle of the big graph, so it'll work. But I was just wondering like.
Yeah, you don't need to label the in a bar graph. You don't need to label the treatments because these treatments are already labeled within the bars there. You just need to label the y-axis.
Instead of bar, I feel like the dot with the error bars.
I'm not sure. That's a tough.
Wait, which graph did you use?
Just like a scatter plot with error bars. Yeah, I don't know if they'll because you weren't supposed to, like I said, you weren't supposed to plot the time. So you weren't supposed to plot like 10 minutes and 24 hours or something like that because then that would be like a ridiculous scale. And so it makes more sense to do a bar graph in that case instead of a scatter plot.
Last question.
Yeah.
So like in the last 2 minutes, then I like drew a new graph in the middle, and then I was like, no, like I don't have time, so I crossed the middle part out. But there was nothing in the middle because like 10 was over here and 24 hours was over there. So do you think they would still read it if I had like corrected
Maybe.
the time?
I think it depends on the reader.
Yeah, like for my time, I just put 10 and I did the zigzag thing and then put 24.
Yeah. Yeah. I don't know. Again, that's going to depend on the reader. They might say it must be a bar graph. I don't know. We'll see. All right, design an experiment using artificial flowers to investigate potential negative effects on increasing caffeine concentrations in nectar on the number of floral visits by bees. Identify the null hypothesis and appropriate control treatment. The predicted results that could be used to reject the null hypothesis. Okay, so design an experiment using artificial flowers. You just got to say something like, I would use 100 artificial flowers that had a sugar water nectar and 100 artificial flowers that had sugar water plus caffeine. Right? Something like that. And then sample them, and you just talk about what you're gonna do, and I'll test the bees in 10-minute intervals or something like that, or record my data and see how long they can remember a flower source or a nectar source.
Wait, if you do like a different molarity, I think one mole very selective.
Of caffeine?
Yeah.
Yeah, that's fine. That's what I'm saying. So one that has caffeine and one that doesn't. Right? The next thing says identify a null hypothesis. The null hypothesis for this should be what? Caffeine has no effect, and anything that we see is due to random chance. Yes. Inappropriate control treatment. What's our control treatment in this one? Yes, no caffeine. And predicted results that can be used to reject the null hypothesis. So if we're rejecting the null hypothesis, and this is actually says, I don't know if this is important, but it does say to investigate the potential negative effects, right? So if it says something like, as the concentration of caffeine increases, the bees die or something like that, or the bee's reproduction is reduced or something. I don't know. Anything that you could say, any result that you got that would be a negative effect that's different than the control. Yeah.
What I did is I had 2 artificial flowers, both with 1 mole of caffeine.
Mm-hmm.
We had 2 bee populations, and after maybe a day of the bees going to the flowers, you increase the concentration of caffeine on one of them to 5 moles,
Mm-hmm.
and see if there's a difference in population.
Yeah, that would be good. And so in this one, it's saying if there was a difference, so you would say if there was a difference between the 1 mole treatment and the 5 mole treatment, then there would be and if there's significant data, that would indicate that you could reject the null hypothesis. Researchers found that nectar with caffeine tends to have lower sugar content than nectar without caffeine. Plants use less energy to produce the caffeine in nectar than they do to produce the sugar in nectar. Propose 1 benefit to plants that produce nectar with caffeine and a lower sugar content. Propose 1 cost to bees. Okay? Benefit to plant is pretty obvious, right? It takes less energy to make the caffeine than it does sugar. And so you have to have less sugar, and so the plant uses less energy and still gets more pollinators, right? That's a benefit. And then propose 1 cost to bees. Why are bees visiting flowers in the first place? No, they don't care about pollinating. To get the nectar to make honey, right? So if they're getting the nectar to make honey and the nectar has less sugar content, that means they make less honey, which means they're not able to feed their offspring and they're not able to survive the winter hibernation months or something like that, right? 10 points possible. 1, 2, 3, 4, 5, 6, 7, 8, 9. I think I missed one of my points. I didn't make a check for this one in here when I said identify the null hypothesis. But it's 10 points possible on that one. 2 for A, 2 for B, 4 for C, and 2 for D. This one was about fires that occur in ecosystems and destroy the above ground vegetation. You've got these 2 compounds, KAR and TMB. They're water soluble. And it has some experiments. Question A, oopsies. Question A says, the researchers made the following claims about the effect of KAR and the effect of TMB on seed germination relative to the control treatment. Provide support using data from figure 1 for each of the researchers' claims. Okay, so KAR alone affects the timing of seed germination. So we find KAR alone, right? And we can see that based on figure 1, we can say that this is A.1. KAR plants, or seeds rather, treatments, germinate how much before? So it's at week 12. At week 12, and control takes how many weeks for the control? Like 18, roughly. I guess maybe they're germinating here. I don't know. About 18, maybe 16, something like that. So that supports that. Then you've got the next question, which was, or the next statement, which is KAR alone affects the percentage of seeds that germinate. Okay, we can see that in the control, only 80% of seeds ended up germinating, but in the KAR treatment, almost 100% germinated. Compared to 80% in the control. And you could also include here, you can see that there's no overlap of those error bars, right?
Will they grade that with a range?
Yeah, of course. Yep. You mean like these 100% and 80% values? Yeah, absolutely. Yeah, it's clearly not 100%, right? It's like 98 or something like that, but maybe you should put 98 instead. I don't know. 97, 98. I don't know. Something like that. Then TMB alone affects the timing of seed germination. TMB alone is here. And so that's like 20 weeks as opposed to, 16 in the control. So TMB treatment germinates at 20 weeks compared to 16 in control. And then for the fourth one, it says TMB alone affects the percentage of seeds that germinate, right? And you can see that it obviously is a lot different, right? So TMB is like 15% versus the 80% in the control. Germination versus 80% in control. 15%, something like that. Yeah, something like that. Okay? So that's A. And so this would be 4 points. 1, 2, 3, 4 points. Then for B, it says, make a claim about the effect of rinsing on the binding of KAR to the receptor in the seed and about the effect of rinsing on the binding of TMB, right? So we can see in this second table that when they had the KAR in treatment group 2 and treatment group 5, there was no change. It was different from the control in both of them, right? So KAR has no change between treatment, was it 2 and 5? Therefore, is not affected by rinsing. something like that. Okay, the TMB, however, the TMB here, here's TMB 3 and 6, right? And so when it's on here, it's different from the control. And then when it's here and there's water in 6, it's the same as the control. And that means that when this gets rinsed away, then it stops inhibiting the seeds, right? So we can say that TMB has a change between treatment 3 and 6. What's happening here? There you go. Between treatment 3 and 6. Therefore, TMB can be rinsed away since the number 6 treatment, or number 6 treatment group, was the same as the control. Or something along those lines. Right, so that's 2 points there. Boom, boom. So we're at 6 now points. We're going to move on to C. Question C says, oh wait, actually this is make a claim and provide support, right? So this one is KR has no change between treatment group 5 and treatment group 2, therefore is not affected by rinsing. So here's our claim, here's our support, here's our support, here's our claim. So that's actually 4 points, right? So this is B is also worth 4 points, so we're up to 8 points now, which means C only has to be worth 2. I want this surface. There we go. So C, there is an intense competition between plants to successfully colonize areas that have been recently cleared. Describe one advantage of KR regulation and one advantage of TMB regulation to plants that live in an ecosystem with regular fires. Okay, so KR regulation is going to cause them to germinate very quickly after a fire. Rapid germination. After a fire, allowing for quick reproduction, right? And then the other one was the TMB. And the TMB, this is the harder part of this question, the TMB, it is going to inhibit the growth, right, until what happens? More water, right? So this waits until the first rainfall after a fire, right? So TMB plants will not germinate until after the first rainfall. Ensuring a proper water source. Something like that. Okay? Question 3. So this is 2 points for that. So 4 points for the first one, 4 points for the second one, 2 points for the third one, yeah.
So, if you mentioned anywhere, like, did you have to mention the water-soluble, like, trait of those two substances?
I don't think so.
And, like, if they both are water-soluble, then how come one you have to wait till the first rainfall and the other one you just, like, have to wait for the smoke and the water?
It's about being rinsed away from their receptors. So, like, whether or not it can be removed from the receptor, like, if it's, because it seems like the KAR, once it hits the receptor, it triggers the germination, and then that's like set in motion. But for the TMB, it's inhibiting it. So it binds to those receptors, and as long as the TMB is bound to those receptors, then it doesn't germinate, and you have to wait until it gets washed away before it can germinate as normal. Does that make sense? All right, so question 3 was about this gibberellin hydroxylase that allows for the enzyme to be converted into active gibberellin. And there's a mutation, and it says the wild type allele. It does say in the question, it says when a pure breeding tall plant is crossed with a pure breeding short plant, the offspring are all tall. But when the F1 plants are crossed, they're 75% tall and 25% short. What is that telling you? What type of inheritance is this? Complete dominance, right? Mendelian complete dominance. So the wild type allele encodes for this enzyme with alanine, a non-polar amino acid at position 229. The mutant allele codes for threonine, a polar amino acid at position 229. The effect of the mutation on the enzyme, describe the effect of the mutation on the enzyme and provide reasoning to support how this mutation results in a short plant phenotype in the homozygous recessive plant. Okay, so describe the effect of the mutation. We're switching a non-polar amino acid for a polar amino acid. So what is that going to do to a protein? What? Changes the shape, because we know that the tertiary structure is the result of the hydrophobic-hydrophilic interactions, and we're changing a nonpolar or a hydrophobic amino acid into a hydrophilic amino acid, which is going to change the shape. Okay, this is a missense mutation. It is going to change the shape of the tertiary structure. I don't know if you would have to say tertiary structure, but I would say, for A, I would say something like missense mutation causes changes in the folding of the tertiary structure due to hydrophobic hydrophilic interactions. Something like that. So let's see. How is that question worded? Describe the effect of the mutation. Okay, so we haven't done that yet. So, what is the effect of this mutation? We'd say that the misfolded protein no longer activates gibberellins. Or can't bind to them or something like that. Right? That's the effect of it.
Why did you know the effect?
Because it says that that when they have the two traits, it says that the plants are short, and then it says that this thing, what it does, it says the GA3 beta hydroxylase is an enzyme that catalyzes the reaction that converts a precursor of a gibberellin to the active form of gibberellin. Right? And so if we change the shape of it, it no longer does that. It no longer activates the gibberellin. Okay, so that is 1 point for making our, describing our change, and then 1 point for the reasoning behind it. Okay, then we got B. B says, oopsies. Using the codon chart provided, predict the change in the codon sequence that resulted in the substitution. Okay, so we're going to go to from ALA to THR. Okay, Okay, and you got to say, okay, well, what's the difference between them? They all have either U, C, A, or G at the end. They all have C as their middle codon. So what's the difference between them? This one has A, and this one has G, right? So we can just say a substitution. Substitution for from G to A in codon or nucleotide number 1. So a point there. And the last point for this question, describe how individuals with one copy or 2 copies of the wild type allele can have the same phenotype, right? So this is a complete dominance, right, situation. And if we have complete dominance, that means that one working copy of the allele or of the gene is enough to produce active gibberellins or something like that. That's what I would say, but you could say probably variations of that. Adobe crashed, hang on. Okay, so that's 4 points for that question. Question 4. All right, question 4 had the food web that looked something like this. And let's go to that real quick. So here's your food web. And it says, based on the food sources indicated in the data table, construct a food web in the template below. So you got to say, okay, well, what do the midges eat? Because that's the first step. We can see the midges are eating stuff. So what do the midges eat? Midges, 100% of their diet is algae, right? So algae goes here. Okay, and then you got to say, okay, what else eats algae? So you go here, and you find algae, and you could say that the caddisflies also eat the algae, right? So that means the caddisflies have to be next. And they eat algae. Then you got to know what eats caddisflies. And it looks like both stoneflies and hellgrammites eat caddisflies. So you got to know which one goes where. And so then you can see that hellgrammites actually eat stoneflies. So the hellgrammites have to be at the top, right? So they're going to be up here. And stoneflies here. And you can see that stoneflies eat caddisflies and hellgrammites eat caddisflies, right?
Like actual things.
Yeah, probably. I don't know. And yeah, and the hellgrammites eat the stoneflies as well. So you end up with something like this. This will get you full credit on that.
How many points would that be?
It's a good question. Let's look at what the other things asked. So construct, and then it says predict whether spraying the fungus, and justify. So this is probably worth 2 points. I would say probably 1 point for putting them in the correct order, and then 1 point for having the correct arrows.
Okay.
Right? 2 points for that. And then for B, it says, in an effort to control the number of midges in an area within an ecosystem, an area within an ecosystem was sprayed with the fungus, yada yada, which significantly decreased the midge population based on the data in the table. Predict whether the spraying of the fungus will have the greatest short-term effect on the population of stoneflies, caddisflies, or hellgrammites. Okay. So, predict and justify. These questions generally have a lot of different answers. So you can say a bunch of different stuff here. And what's up?
I just came back to say goodbye.
Did you take a test yet? Your
Yep.
exam? Isn't that too bad?
It had a lot of circuits on it.
Hmm.
Circuits and then not much coding, but it was mainly circuits and physics.
Cool. All right. So, you could make a lot of claims here. Immediately, I'm drawn to, all right, look, this one eats 90% midges. It's 90% of its diet. And so that seems pretty bad if the midges die out, because 90% of its diet is going to die out. So I would say stoneflies are the most affected because 90% of their diet is midges. You could probably also get away with saying caddisflies would be most affected, because the caddisflies are the food source of the stoneflies and the hellgrammites, right? But midges were also food sources for those things, and so if you kill off the midges, then the caddisflies are the only things that these things have to eat, and so their population is probably going to go down. But that might be offset by the fact that now they don't have to compete with the midges for the algae. I don't know. Basically, I think you could have said a lot of things here as long as you justify them. What's up?
So is there an arrow that goes from algae to midges? Or no? Like for the way the energy is
This way?
Yeah.
Yeah, there was a provided error, or provided arrow.
Okay. I didn't know if those arrows had counted, so I just had them.
Yep. Okay. 4 points for that one. Question 5. This thing is a freshwater photosynthetic cyanobacterium. When temperatures increase, the nutrients are readily available in its pond habitat. This cyanobacterium undergoes rapid cell division and forms an extremely large visible mass of cells called an algal bloom. The cyanobacteria has a short lifespan and is decomposed by aerobic bacteria. Identify the metabolic pathway and the organism that is primarily responsible for the change in oxygen level between 1 and 2 and 3 and 4. So this is a 4-point question. It's just identify. So you're going to get points on this one for saying, this is question 5. You're going to get points for saying between, what was it, 1 and 2? 1 and 2. The cyanobacteria. It says the organism. So this is interesting to me. So it says the organism that's responsible, and then up here it's labeled as cyanobacteria, but down here it gives you the actual name of it. So I don't know if they want you to say cyanobacteria or they want you to say this actual genus and species of it.
I said the name and then I was like the cyanobacteria.
Yeah, I think that if you said both, you're definitely fine. But I think that if you said either, you're probably also fine, right? So I would say, like, between 1 and 2, cyanobacteria were performing photosynthesis. Increasing O2 levels. And then we could say that between between 3 and 4 decomposers are performing cellular respiration. Decreasing O2 levels. Something like that. Okay, and then so that's cyanobacteria, photosynthesis, decomposers, cellular respiration. If I was a grader, an AP grader, this would be the question that I would want to grade. It's just like 4 words that you had to say. If you said those 4 words, that's all the points.
Yeah.
All right, so the next one was question 6. Question 6 was about this comet assay. And they say that you can detect the DNA breakage, double-strand breakage. Okay, it says to explain the movement of DNA fragments in the comet assay, explain one property of DNA and provide reasoning to support how the property contributes to the movement during the comet assay technique. Okay, well, we know that DNA is negatively charged due to all the phosphate groups, right? And so if it's negatively charged, which end of the assay is it going to move towards? Positive end. So we say that negatively charged DNA is attracted towards the positive electrode. And I think that gets you the points. What did they say? Identify the property of DNA, negatively charged. Provide reasoning to support how the property contributes to the movement. So we'd say, like I said, that it moves here because it's moving towards the positive electrode. Okay. It says, I don't know if you need to mention this, but it makes sense for the style of question, even though they didn't directly ask it. So I hope since they didn't directly ask it, they won't want you to say it. But the reason why the tail is longer in when there's more damage is because we know that shorter fragments of DNA are going to travel further amounts of distance in the agarose gel, right? And so, if you just have a little bit of damage, right, it's just going to go like that. But if you have a lot of damage, it'll be like that, right? Because the short fragments are going to move a lot. If there's a lot of short fragments, they'll be pulled towards the positive end more.
I don't remember knowing that DNA is negatively charged.
We talked about it when we did DNA gel electrophoresis.
Wait, why is it?
That it always it's negatively charged due to the phosphate groups.
So phosphate is negative because on the side of that of the chart.
PO4 3 minus is a phosphate group. That's from chemistry.
And that's how gel electrophoresis works.
That's how gel electrophoresis works. Yeah, the DNA is negatively charged, and when you put in you put in an electric current, that since it's negatively charged, it's going to move towards the positive end.
Okay.
It's going to be repelled from the negative end and pushed towards the positive. Repelled from negative, attracted to positive.
Okay, so then you can see the amino acid distribution.
Yeah, well, in gel electrophoresis, you're seeing the distribution of fragments, right? And the smaller fragments get pulled further towards the positive end. But in this, you're just looking at like the length of the tail. You're not worried specifically about the number of nucleotides in each fragment or anything like that. So yeah, that was 2 points for that. It says, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions. Predict the likely results. No tail at all. You wouldn't see any tail because this is causing substitutions, and this thing only detects breaks in the DNA. Right? And since it's only looking at breaks, there'd be no comet at all, no tail formed at all. Question 7. So that was 3 points possible on that one. Question 7 also has 3 points possible on it. Many species of bacteria grow in the mouths of animals and can form biofilms on the teeth, or on teeth. Within plaque, the outer layers contain high levels of oxygen, and the layers closest to the tooth contain low levels of oxygen. The surface of the tooth is covered on a hard layer of enamel, which can be dissolved under acidic conditions. When the enamel breaks down, the bacteria in plaque can extract nutrients from the tooth and cause cavities. Okay. Certain types of bacteria, this S. mutans, thrive in the innermost anaerobic layers of the plaque and are associated with cavities. Other types of bacteria, S. sanguinis, compete with S. mutans, but are unable to thrive in acidic conditions or acidic environments. Identify the biochemical pathway of S. mutans uses for metabolizing sugar and describe how the pathway contributes to the low pH in the inner layers of plaque. Okay? Where does the S. mutans live? It thrives in the innermost layers, innermost anaerobic layers, right? So what kind of metabolism would you have in an anaerobic environment? Fermentation, specifically lactic acid fermentation. Okay, I don't know if they specifically want you to mention glycolysis here, because it does say, you know, what metabolic pathway do they use to break down the sugar. I would assume lactic acid fermentation is enough, but they are using glycolysis only to break down the sugars here. And why does the pH drop then?
Because you got acidic lactic.
Yeah, production of lactic acid.
You don't have to make complete sentences with these, right?
You should try.
You should try?
Yeah.
Okay.
Yeah, so yeah, my answers here are not what you would want. They'd probably get you points, but they wouldn't be happy about it.
Okay.
You should try to do full sentences. Normal toothbrushing effectively removes much of the plaque from the flat surfaces of teeth, but cannot reach the surfaces between the teeth. Many commercial toothpastes contain alkaline components which raise the pH of the mouth. Okay. Predict how the population sizes of S. mutans and S. sanguinis in the bacterial community in the plaque between the teeth are likely to change with these when these toothpastes are used. Okay, well, it doesn't really do anything to the plaque layer, right? But it does say it raises the overall pH of the mouth. So this one's tricky to me because within the plaque, the outer layers have high levels of oxygen, lowers. The surface of the tooth scope, which can be dissolved under acidic conditions. I think what they want you to say here is that the population of S. sanguinis or whatever. What is it? S-A-N-G-U-I-N-I-S. Population increases due to less acidic conditions. S. mutans decreases due to lack of competitive advantage, something like that. Due to pH change.
So, more acidic conditions?
Less acidic conditions. Something like that, because the toothpaste is gonna make the mouth less acidic. It's gonna increase.
Alkaline.
Yeah. So there's your 3 points for this one, right? So 1, 2, 3 points. And then last question, question 8. It's about estrogens. Estrogens are small hydrophobic lipid hormones that promote cell division and the development of reproductive structures in mammals. Estrogens passively diffuse across the plasma membrane and bind to their receptor proteins in the cytoplasm of target cells. Describe one characteristic of the plasma membrane that allows estrogens to passively cross the membrane. Okay, what do we know about the plasma membrane? Phospholipid bilayer, and the majority of the phospholipid bilayer are those fatty acid tails, which are nonpolar, right? So we'd say, put them on the stack, actually. Yep. Nonpolar fatty acid tails allow nonpolar substances to diffuse through them. Okay. 1 point. In a laboratory experiment, researcher generates antibodies that bind to purified estrogen receptors extracted from cells. The researchers use antibodies in an attempt to treat estrogen-dependent cancers, but finds that the treatment is ineffective. Explain the ineffectiveness of antibodies for treating estrogen-dependent cancers. Okay. So this one seems pretty easy. What do we know about the receptors for these estrogens? Where are they? They're on the inside of the cell, right? So the estrogen receptors are found on the inside of the cell. And where are proteins? Are those able to diffuse through the membrane? No. Are antibodies found inside cells? No, they're part of the humoral immune response, right? Which means they're only outside the cell. So we'd say that the proteins stay outside the cell. Due to polarity, and size. Actually, we should probably say protein antibodies. So protein antibodies stay outside the cell due to polarity and size. Estrogen receptors are found inside the cell. Treatment ineffective because antibody never meets receptor. What's up?
Okay, so the estrogen receptors are on proteins, and proteins can't link to this, right?
Estrogen receptors are what?
Are on they're on the proteins in the cell.
They're probably located on the nucleus. They're bound to the nucleus.
Okay, and then how do you know the polarity of the antibodies?
Because they're made out of proteins. Remember, there's like two types of, when we talked about hormones, we said there were two types of hormones. There's fat soluble and water soluble. And the water soluble ones that can't go in, those are the proteins. And so you should be able to say like, okay, proteins then are too large and too polar to go inside of a cell without any sort of facilitated diffusion. You could also say something like antibodies